Inside every single one of your trillions of cells, right this very second, an absolutely incredible story is unfolding. It's the journey of how a microscopic blueprint dictates, well, everything about you. So today, we're going to trace that amazing information highway from DNA to RNA and all the way to protein. Let's get right into it. Okay, so it all starts right here, inside the nucleus of the cell. The best way to think about it is like a master library, right? A super secure vault that holds the complete architectural plans for building an entire organism. It's just mind-boggling that so much information is packed into a space that tiny. And that's the big question, isn't it? That's the mystery we're unraveling in this explainer. How on earth does this system manage such a colossal amount of data with such perfect precision, second by second? Well, the answer is just a masterpiece of molecular engineering. So our first hurdle is basically a packaging problem. Get this, if you were to stretch out the DNA from just one of your cells, it would be about 2 meters long. So how in the world do you cram that into a nucleus that's just a few micrometers across? The cell's solution is honestly brilliant. It's something called chromatin. Imagine your DNA molecule is a long piece of thread. The cell winds that thread super tightly around these tiny protein spools called histones. This whole DNA and histone unit is called a nucleosome, and it creates the structure that looks kind of like beads on a string, allowing for just incredible compaction. But here's the cool part. This library has different access levels. On one side, you've got euchromatin. It's all loosely packed, kind of like an open book on a desk. This is the open for business section, where genes are actively being read. But then you've got heterochromatin, which is coiled up super tight and locked away. These genes are in deep storage. They're silenced, turned off. Okay, so we've figured out how to file away the blueprint. But what happens when a cell needs to divide and make a new one? Well, you're going to need a perfect copy of the entire library. And that brings us to the really high-stakes process of replication. And this is just a non-negotiable rule of life. Before a cell can divide, the entire genome, the whole blueprint, has to be duplicated with almost perfect accuracy. I mean, a single mistake can have massive consequences for that new cell. The whole process kicks off with an enzyme called helicase, and its job is simple but absolutely essential. It's the unzipper. It moves along the DNA double helix, unwinding and separating the two strands. This exposes that sequence of letters so they can finally be read and copied. Now, once the strands are separated, the real star of the show arrives, DNA polymerase. This enzyme is the master builder. It zips along the template strand, reading the code and grabbing the right building blocks to create a brand new matching strand. And what's amazing is that it's unbelievably fast, and it even proofreads its own work as it goes. So here's a little quirk. One of the new strands gets built in one continuous piece, but the other one, it's called the lagging strand, has to be built in all these little chunks. That's where DNA ligase comes in. It's basically the molecular glue. It comes along and seals all the gaps between these fragments, creating one seamless new DNA strand. But even with all that proofreading, mistakes can still happen. And sometimes DNA gets damaged from the outside, like by UV light from the sun. The cell actually has an entire toolkit of repair crews. For example, a system called nucleotide excision repair can literally snip out a section that's been damaged by UV rays and then replace it with the correct sequence. This isn't just routine maintenance. It's a critical defense against mutations that could lead to diseases like cancer. All right, so now we have a perfectly stored and perfectly replicated master blueprint. But think about it. You would never take the original priceless architectural plans out to a messy construction site, would you? Of course not. You'd make a working copy. And in the cell, that copy is made of RNA. And that is such a crucial point. The precious DNA blueprint stays safe and sound, protected inside the nucleus. To actually use an instruction, the cell creates a temporary, disposable copy of just a single gene. This working copy, this little memo, is called messenger RNA, or as you've probably heard, mRNA. But this first RNA copy, it's not quite ready to go just yet. It has to go through a little bit of processing first. There are 3 key steps. First, a protective cap is added to one end, kind of like a hard hat. Then, a long stabilizing tail is attached to the other end. And finally, a process called splicing happens, where all the non-coding junk sections, called introns, are precisely cut out, and the important bits, the exons, are stitched together. So now, with all its modifications complete, our mature mRNA memo leaves the nucleus and travels out into the cytoplasm, which is basically the cell's factory floor.
This is where the instructions on that memo are finally going to be used to build the actual product, a protein. And this whole process is called translation. The factory itself is this amazing molecular machine called a ribosome. It literally clamps onto the mRNA strand and starts to read the genetic code 3 letters at a time. Each of these 3-letter words, called a codon, specifies one of the 20 different amino acids, which are the building blocks of all proteins.
And as you can see here, this whole assembly line works in 3 main stages. First up is initiation. That's where the ribosome finds the start signal on the mRNA and gets set up. Next is elongation. This is where little courier molecules called tRNA deliver the exact right amino acid, one by one, and the ribosome links them all into a growing chain. And finally, you have termination. The ribosome hits a stop signal on the memo and, poof, the brand new finished protein is released.
This entire system is just breathtakingly precise. But, it's a biological process, and it's not 100% perfect. So what happens when there's a typo, a mutation, in that original DNA blueprint? Just think about that for a second. The human genome has about 3 billion letters of code, and a single tiny error, one wrong letter in all of that, can have consequences that range from completely harmless to absolutely devastating. So let's compare 2 different outcomes from just one little letter change. In what's called a silent mutation, you basically get lucky. Because of a little quirk in the genetic code, the new 3-letter codon just happens to code for the exact same amino acid. No harm, no foul. Phew! But a nonsense mutation, well, that's a showstopper. It changes a codon that's supposed to add an amino acid into a stop signal. Production just grinds to a halt, creating a useless, shortened protein. And then there's something even more disruptive, called a frame shift mutation. This is when a letter is either added or deleted entirely. Because the code is read in groups of 3, this one tiny error shifts the entire reading frame for every single codon that comes after it. It's like reading a sentence where one letter is missing at the beginning. The rest of it is just complete gibberish. The result is a totally garbled message. And these blueprint flaws, they aren't just abstract ideas. They are the root cause of so many genetic diseases. A single missense mutation, that's where one amino acid gets swapped for another, is what causes sickle cell disease. That one tiny change makes hemoglobin molecules clump together, distorting red blood cells into that classic sickle shape. And as we just saw, those more drastic frame shift mutations are behind diseases like Tay-Sachs and cystic fibrosis. It all comes back to a tiny error in the code. So when you step back and look at the whole thing, this elegant, intricate flow of information from DNA to RNA to protein, it really is the central principle of life itself. It is happening constantly in almost every cell in your body at this very moment. We've followed this incredible journey from the blueprint to the final product, and we've seen the massive impact of even the smallest flaws. It's a system of unbelievable complexity and power. And that leaves us with one final, pretty profound question to think about. If this is the blueprint for life, what happens when we learn how to edit it?